Hello, everyone, and thank you for joining us for the second session of our two-part webinar series with Jason. My name is Odine Alvarez, and I'm a soil health educator with the Soil Health Institute. My colleagues Shannon Moeller with the Soil Health Institute and Olivia Cayuet with the University of Missouri Center for Regenerative Agriculture are also in this call, providing technical support. And the three of us support the education component for Farmers for Soil Health. The purpose of today's webinar is to take a deeper look at how soil health measurements are integrated into the Soil Health Institute's benchmarking approach, which is an approach designed to assess how healthy a soil is today and how it can improve over time. Today's webinar is supported by Farmers for Soil Health, or FSH. The initiative is one of many USDA-funded advancing markets for producer projects. And the FSH is a collaborative initiative of commodity organizations, includes the United Soybean Board, the National Corn Grower Association, and the National Pork Board, with a goal to increase cover crop adoption. This specific project provides an incentive to farmers who are new to planting cover crops or who have been planting cover crops for many years. The state level commodity organizations and conservation professionals work across 20 states, stretching from the Dakotas to the East Coast to provide local technical assistance and outreach programming. FSH has also developed a website that has a lot of resources and information, and you can learn more about it at farmersforsoilhealth.com. Lastly, a few more items about today's event. This presentation is being recorded and an email will be sent with a link to the webinar recorded, posted to the Soil Health Institute's YouTube channel, along with a final feedback survey. And we'll end with a Q&A that will be moderated by Shannon. So as questions come up, please feel free to put them in the Q&A, and you can upvote questions that you'd like to prioritize as we cover them live. And if you run into any technical issues, please chat the webinar co-hosts. We'll work with you to resolve those. And if you like captions, those can be enabled by clicking the three dots on your toolbar and navigating to captions. And be sure to hang out until the end for information related to Certified Crop Advisor Continuing Education Unit for attending this webinar. And with all of that being said, I'm excited to now welcome Dr. Jason Ackerson again for the second part of this webinar series, and I'll let you introduce yourself and take it away.
Great. Thanks, Odine. And thank you to the Farmers for Soil Health team for, helping host this webinar and put this on. I think this is a really exciting topic. So today we'll be talking about soil health benchmarking in, the context of this two-part webinar series on soil health measurements and benchmarking. So, we're going to start with a bit of a recap of what we discussed last week. So if you missed out on last week's recording, either you missed the webinar or didn't have a chance to get up to date on that recording, we'll kind of cover a little bit of the ground we covered last week. So hopefully everyone's starting from a relatively even playing field. So last week we talked to a large degree about how we measure soil health. So really focusing on the nitty-gritty details around what measurements we recommend at the Soil Health Institute for measuring and quantifying soil health. And what we didn't really touch on is, interpreting those numbers. And that's really the goal of today's webinar, is to talk about soil health benchmarking, which is the tool we've developed to help farmers and advisors understand those soil health indicators and put them in the proper context so we can make good management decisions about soil health. So, like I said, we're going to start with a bit of a refresher on what we talked about last week, and then we'll dive into, some of the details around benchmarking with the focus kind of big picture on the philosophy of why we've developed benchmarking the way we have and how those considerations affect some of the outputs from benchmarking. So first, who is the Soil Health Institute? I'm sure many of you have seen this slide or a similar slide before, but the Soil Health Institute is a global nonprofit with the mission to safeguard and enhance the vitality and productivity of soils through scientific research and advancement. Essentially, that is to say, like, we're a small nonprofit full of people who care passionately about soil. And one of the things we've really worked really hard on over the last, 7 years, 10 years of the Soil Health Institute really is focusing on this question of what do we measure when we want to measure soil health and how do we make sense of those data? And so hopefully in this webinar series, we'll get to touch on both of those pieces. Like last week, we talked about what do we measure, and hopefully this week we'll give you some context for how to make sense of those measurements. But why do we care about soil health in the first place? And for me, it all comes down to soil function. We want soils to function effectively as part of, effective, productive agroecosystems. So the picture you're looking at here, which I'm sure is familiar to anyone who's seen the Soil Health Institute talk, I think it's a great picture that really illustrates the effect that soil health has on agroecosystems. So on the right-hand side of the screen, we're seeing a soybean field that's managed under soil health. Soil health's on the right, right? So this is a field that has focused on reducing soil disturbance by transitioning to no-till. Occasionally there's cover crops, so they're trying to maximize living roots, and they're keeping the soil covered and armored. So when you look at that soil, you see tons of crop residue that's keeping that soil insulated from any physical wear and tear. On the left-hand side of the screen, we're seeing a more baseline or conventional management system where those soil health practices aren't employed. And that has drastic consequences for how that soil functions as an agroecosystem. Um, on the left-hand side, we can see there's lots of water and ponding on the soil surface. So a recent rain event has caused a bunch of water to sort of puddle up on the surface, causing surface sealing. And that has cascading effects to the agricultural productivity of that system, right? If you look, you're seeing poor stand emergence in these soybeans. There's gaps. In a larger scale, this might require, replanting operations, which is costly for growers, and lowers the productivity of those soils. On the right, the soil health soil, really great stand establishment. We don't see any water puddling or ponding on the surface, and we see no evidence of surface sealing or surface crusting. So the soil is functioning a lot better. And that's really when we think about soil health, we're thinking about things that indicate effective soil functioning. And we've talked about some of these functions on the previous slide, but just to sort of recap, right, we hit heavily on water infiltration and retention. That's a really important physical function of the soil. And we talked about the impacts that that has on crop production and biomass generation and biomass provisioning, right? Soils are essential parts of terrestrial ecosystems when it comes to, supporting plant growth. And that also relates to the biogeochemical cycling of carbon and nutrients in these systems. Healthy soils function better with regards to how they store and move carbon around, and then the associated nutrients that are related to carbon, organic nitrogen, and then some of our inorganic fertility can be the movement of those elements is affected by the soils and its function, which really, is an expression of two components. We've talked about in the last couple slides on how management might affect soil functioning, but also, soil functioning is constrained by the inherent properties of the soils. You can't make soils in Texas function the same as soils in Illinois or Iowa. They're different ecosystems. The soils have different constraints, and we have to understand the effect of those constraints when we're thinking about soil health, because ultimately the soil health indicators we'll be discussing today, and we discussed at length last week, are the product of the management plus the soil environment. So those inherent properties plus the management gives us the expression of soil health that we'll measure and the soil functions that we think that those measurements represent. But ultimately, measuring soil health is about thinking about functions and thinking about how productive or healthy the soils are. So we can measure soil health to identify potential problems with soil functions. It's an important part of conservation planning, and we've seen with the new USDA regenerative pilot program that soil health testing under SEMA 216 is part of that conservation planning process. So it's great to see that recognition at the federal level. But also, measuring soil health helps us track and understand how management practices on our farms are affecting soil health and soil function. And lastly, our CSO likes to say this, and I think it's a great line, if you treasure it, you measure it. We care deeply about how soils function as part of, our agricultural systems. We should also understand and measure how well they're functioning. So in order to sort of get to the meat of what do we measure in when we talk about Soil Health Institute conducted a large continental study, which we refer to the North American Project to Evaluate Soil Health Measurements, or NAPSHM for short. So if you hear me say NAPSHM, that's really what I'm talking about, is this big study where SHI scientists went to 124 long-term experimental trials across North America, and we measured everything you could think about when it comes to measuring or quantifying soil health. And really the goal of that work was to say, okay, here are the 30 things we could measure when we want to measure soil health. What are the ones that we think are most effective and scalable? So from those data, we applied a set of filters or selection criteria to the NAPSHM data. So we looked at, we wanted to select only measurements that were reflective of soil health. So we threw out anything that was an indicator of the inherent soil properties. But then we picked indicators that were responsive to management. So when you adopted no-till, did the indicator change? When you adopted cover crops, did the indicator change? Right? Were these indicators reflecting the changes in soil management that was part of the experimental setups at each of these long-term trials? And then our last two filters really focused on the scalability and redundancy of those measurements. We didn't want to take measurements that told us the same story, so we didn't want redundant measurements. And we wanted measurements that could be done at commercial labs at scale, so they could be part of, large scale soil testing programs. So from that NAPSHM data and that filtering criteria, we got to what we call the essential soil health indicators. So we have three measurements. That would be soil organic carbon, which is a measurement of the total amount of organic carbon in the soil, which is the basis of the microbial food chain. It has chemical properties that help the soil retain structure and nutrients and absorb water. We also measure a measurement of the biological activity of the soil through carbon mineralization potential, which tells us how much CO2 is being emitted by the microbes in the soil. So more CO2 emitted by the microbes means we have more active, healthy soil microbiological communities. And then lastly, we have some physical measurements through aggregate stability, which is a measurement of how the physical structure of the soil maintains its shape when it gets wet. And then we use a pedotransfer function to predict the plant available water, which tells us how much water is stored in the soil. So with those four measurements, we think we have a pretty good understanding of, a good picture of what soil health might be in any given soil. But that really doesn't get us to, a functional outcome for a grower or an advisor who might want to answer the question, how healthy is my soil? So the rest of today's talk, we're going to, focus in on what we call soil health benchmarking, which is a way of taking those essential soil health indicators and making sense of them and understanding, how healthy is the soil based on what I've measured with those essential indicators. And it's really hard to talk about soil health without drawing an analog to human health. So, when you go to the doctor takes a bunch of measurements to try to give a picture of your health. These might be things like blood pressure, temperature, heart rate, body mass. We measure a bunch of things that tell us how healthy we are. We do the same thing with soil, right? We measure the organic carbon concentration, we measure the aggregate stability, we measure the carbon mineralization potential, and those give us a good picture of how healthy a soil is. So, sort of drawing on that soil health, human health analogy, um, we'll do a little thought experiment. So, we're going to compare two completely random individuals. On the left is Jason. He works at a desk all day, does not get to exercise as much as he would like, cannot dunk a basketball, and he weighs 210 pounds. On the right, we have this gentleman named LeBron. He, in his spare time, designs tennis shoes. He's a four-time NBA MVP, and he weighs 250 pounds. So, based on the information you've heard today, who is the healthier individual, Jason or LeBron? So you might look at their weights and you might say, "Oh, LeBron, he weighs more than Jason, so he must be less healthy, right? He's heavier." But we know, that's a fiction. And when it comes to interpreting human health, we know the story is more nuanced and making those comparisons is a relative type of comparison. So body mass alone, anyone can understand that is a poor indicator of soil health because taller individuals by, nature of having more bones are heavier people. So doctors have developed this index called the body mass index, which is essentially a ratio of your body mass, your weight to your height. So it helps try to account for the effect of your height on your overall health. So higher body mass indices are correlated with obesity and then all of the negative consequences of obesity. So the two ways of there's one way of changing your body mass. You can't make yourself taller, or sorry, excuse me. There's one way of changing your body mass index. You can't make yourself taller or shorter, but you can get heavier or lighter. And so body mass index is really only affected by the dynamic property of someone's weight at the time of measurement. But we need the inherent property, that static property of their height, to make heads or tails of what that measurement is. So going back to our analogy with our two randomly selected individuals. Jason has a height of 6'2" weight of 210 pounds, which gives him a body mass of around 207, or sorry, 27. LeBron, turns out he's a lot taller than Jason. He's 6'9", close to 7 feet, and he weighs 250 pounds. So his body mass is just slightly less, body mass index is slightly less than Jason's at 2.68. So good news for Jason. He's almost as healthy as LeBron James is in terms of his body mass index. And so to draw the parallel here with human health and soil health again, we need to understand how those inherent characteristics or properties affect the health of an individual or of a soil in order to make the right kinds of conclusions around those health measurements. So in humans, we need to understand someone's age, their sex, their height, their genetic history. Those are all inherent or static properties that help us interpret whether or not their health indicators are good or bad. So with their body weight, their blood pressure, their heart rate, their temperature, all might be affected by those inherent properties. And it's important to understand those inherent properties to get a full picture of the human health. And the same story is true for soils. So inherent properties like the climate, the texture of the soil, the drainage class, the mineralogy, topography, all of those things can have an impact on the potential ranges of our soil health indicators, and they need to be accounted for in order to really draw good conclusions around how, soil health is indicated by those essential soil health indicators. So I think carbon is a really great example of this. So the graph you're looking at here is a prediction of the amount of soil organic carbon in the topsoil of soils for North America based on remote sensing data. So places that are darker on the map have higher carbon concentrations than places that are lower on the map. So the yellower sites are lower in carbon. And you can see, some really clear patterns geographically, places with high carbon concentrations in their soils. The Midwest, those corn, soybean states have lots of carbon. If you move further south or southeast specifically, as soils get more dry, hotter, tend to have less carbon. So this is showing us, really visually how the inherent properties of climate are impacting how much carbon is stored in the soil. And similarly to our body mass index analogy, we wouldn't want to compare someone who's 6'2", who lives in Texas, to someone who's 6'9", who might be in Iowa. Their carbon concentrations are going to be very different just based on the climate that they're in, not necessarily how they've managed their bodies, right? Their health might not be similar. So we need to account for those climatic conditions when we're making soil health comparisons. A similar story with all of our soil health indicators, but I thought this was a nice illustration of that as well for our aggregate stability measurements. So in this case, you're looking at data from um, aggregate stability measured with SHI's SLAKES app. So this is aggregate stability through image analysis, where we put an aggregate in water, it disperses or slakes, and we measure the amount of dispersion as an indicator of aggregate stability. And we see that in general terms, these are data from all over North America. We see that as texture increases, that aggregate stability index tends to go down. Similarly, as precipitation increases, the aggregate stability index tends to go up. So wetter soils have a higher aggregate stability index than our drier soils. It's important to note here, we often think about sandy soils as having a lower aggregate stability than clay soils. And that is true in a sort of relative sense of the structure of those units is tends to be higher. When we look at this data from our slaking indicator, we often see that because those sandy soils with low clay contents slake so quickly, we don't see a high degree of slaking in those sandy soils. So the clay content here can be a little misleading, but it's a good reminder that when we're comparing soil health, it's more important to look across the vertical axis here and compare soils with similar clay contents. So we wouldn't want to compare the aggregate stability of a soil with a clay content of 60% to the aggregate stability of a soil with 20% clay content. And that's the fundamental component of soil health benchmarking that we'll kind of unpack in the next couple slides. Which brings us to soil health benchmarking. So we've talked a lot about these abstract concepts of inherent soil properties and dynamic soil properties and soil health indicators. How does that all fit together in a way that allows us to make meaningful interpretations of soil health measurements? And that's where the Soil Health Institute has sort of landed on this procedure that we call soil health benchmarking. And really, the goal is to help us know what the likely ranges of any soil health indicator are for a specific soil in a specific geography under a specific management regime. So there's a lot of specificity in that, but really, it's a long-winded way of saying we want to account for how the inherent properties, those things like texture, climate, topography, the pedological environment that that soil was formed in, as well as the management regime that that soil is managed under, in concert to understand how that soil health indicator might change in response to management. So you'll often see our benchmarking graphs illustrated as a set of like three colored bars, and each of those colored bars represents, a canonical management regime. So in the sort of yellow color, and this is consistent through the rest of this talk, is what we refer to as baseline management. So these would be soils managed. Often people refer to this as business as usual, but these would be soils managed without any extra considerations for improving soil health. In the blue bars, you'll always see data representing our soil health management systems. So these are row crop systems still, but they are now managed with the goal of maximizing or improving soil health, either through adoption of no-till practices, integration of cover crops, methods of improving soil armor. All of those sort of fundamental soil health practices are being expressed in some of the blue data. And then lastly, you'll see data represented in the dark green color, which represents what we call a reference site. So this is a site where soil health principles are, turned all the way up to 11. We have continuous living roots all year. The soil is always covered, and disturbance is basically zero. So in most of our data, these reference sites are places that are under some sort of perennial vegetation, pastures, reclaimed prairies, almond orchards, fence rows where we have perennial grasses, CRP land, places where the soil health principles are maximized. So that's a kind of abstract way of talking about benchmarking, but to the rest of the talk, I'm going to walk you through kind of the steps that we go through in order to develop some benchmarking outputs. So it all starts with soil sampling. The Soil Health Institute does a bunch of soil sampling with cooperating farmers. but it all often starts by us identifying a region or geography that we want to target or focus in on. Then within that geography, we look at the soils maps, we identify what the most important soils are, and we focus on sampling those. And then we go out and we collect soil samples. We want to collect multiple samples under various management regimes, like I showed you on the last slide. Then we move into the data analysis part, which for nerds like myself is often some of the most fun. We fit a statistical model, and the goal of that model is to account for the effects of those inherent soil properties and management on our soil health indicators. And then we use those models to understand like what are the reasonable ranges of our essential soil health indicators under various management scenarios. And then we get to maybe the most practical part, which is reporting, sharing the results of that work, either through a regional report or a farmer report that kind of either gives a high level overview in the case of regional reports, or a specific, field level report on an individual sampling basis. So for the rest of the talk, I'm going to walk you through each of these steps in a little bit more detail to give you an understanding of how we go about benchmarking, and then we'll talk about some of the implications or outputs from the benchmarking process. So, like I mentioned, it all starts with soil sampling. So we want to target soils that are the most representative of row crop acres in any given geography. And to do that, we've developed what we call soil health sampling groups. And this is a way of grouping soils that have similar textures and drainage classes, we expect will have a similar relationship to management, or they'll respond the same to management and therefore have similar potential soil health. So we want to group soils in such a way that we can try to, minimize how many unique types of soil we're sampling on the landscape. So what you're looking at here is a graph of North America, where we've taken existing soils data predominantly from the USDA Soil Survey or the SURGO database, and we've taken the data about those soil types and we've applied our soil health sampling group concept to them and then grouped them into the sampling groups within each of the major land resource areas in CONUS. And so the background color of this map represents the unique soil health sampling groups, as described in the legend here. So the sort of orange colors are the clayey textured soils, blue colors are the silty soils, the green colors are sort of the fine loamy, the yellow or the coarse loamy, or sorry, the coarse silty, and then we have coarse loamy and sandy soils in the sort of orange and pink colors respectively. And then the how saturated that color is represents the drainage class. And the ultimate, goal of this soil health sampling groups approach is to minimize how many unique soils we're trying to sample. So if we tried to look at all of the unique map units in the US, it's somewhere around 23,000 soil types we try to have to categorize. And we minimize that significantly to just over 4,500 soil health sampling groups. It's a significant reduction in how many types of soils we're looking at. So once we've, identified a region, we've taken it and looked at soil health sampling groups, then we send folks out into the field to collect soil samples. And their goal is to, within our target soil health sampling groups, collect soil samples from a range of farms under different management regimes. So we try to target farms under that baseline scenario where we don't use any soil health practices. They try to find growers or farms where soil health management is being employed, row crops with cover crops or no-till. These are places where we think soil health is improved over the baseline sites. And then they also focus in on those reference sites. These are perennial systems where soil health principles are being maximized, and we think that will likely have the highest level of soil health indicators. So a little bit more on those reference sites, you'll see in all of the graphs we'll show later, they tend to have the highest levels of soil health indicators, and these are probably the optimal state where indicators are maximized due to having the most rigorous implementation of NRCS soil health principles of minimizing disturbance, maximizing roots, maximizing soil cover, and maximizing biodiversity. And the goal of the references that you'll see is really to kind of understand what the upper limit of soil health indicators are in any given soil as and provide that sort of background of how soil health indicators change in response to inherent properties when we're not disturbing the soil through row crop agriculture. So, soil health sampling groups, we've identified which soils to collect. We've sent folks out to the field to collect soil samples on a bunch of different managements, including reference sites. And now we've got this pile of data, and what do we do with it? Um, I'm not going to get super into the weeds on how we actually fit or do any of the statistical modeling here, but what I want you to get a sense for is how the models relate these two concepts of inherent soil properties and essential soil health indicators. So the goal of all of our benchmarking modeling work is to develop a statistical framework that lets us understand the relationships between our essential soil health indicators and those inherent soil properties. So we can do the same thing we did in that BMI calculation, where we want to account for, the natural changes in a soil's potential to have various levels of soil health indicators so we can make good inferences on measurements on real soils. So what you're looking at here is the output of one of these soil health benchmarking models where we're showing three separate lines for each of our management scenarios. So this model is comparing wet aggregate stability as a function of clay content. And there's other indicators wrapped up in this model, but we can't visualize them on one graph. So I've simplified it here for one graph of showing how aggregate stability and clay content are related for this specific geography and how management affects that. So you'll notice the baseline systems in gold, those are the systems without soil health practices, have the lowest on average aggregate stability, indicated by that solid line being on the lower side of all three of our management systems. At the very top are our reference sites. So again, these are the sites where soil health principles are maximized, and we think soil health indicators are going to be at their highest levels. So that line is at the highest part of our graph. And then somewhere between there is the blue line, which represents soils or sites managed for improved soil health. And so this line is representing the average sort of outcome of aggregate stability as a function of clay content for those soil health sites. So this is a long-winded way of saying now we think we have a way of doing the same thing we did with that body mass index, but now instead of comparing human weights, we're comparing aggregate stability and how clay content, affects the inherent levels of aggregate stability for various soils. So in practice, we might ask the question, okay, I have a farmer. We've taken an aggregate stability sample from this region. They have a clay content of 25% and an aggregate stability of 0.65. How healthy is their soil? So soil health benchmarking helps us answer that question. So we start by looking at our models and saying, okay, I want to look at what the models are predicting for our soil health indicators, given the inherent property of 25% clay. So we think about this as sort of a vertical line or a slice through our models, represented by the dashed green line on the graph. And then everywhere where that line intersects our modeled results, we can think about, capturing some of that output. So in the case of our reference sites indicated here by the green line, we can pull out from that model the predicted ranges of aggregate stability for all soils in this region that have a clay content of 25%. And that's represented here by the vertical bar. So that represents, what our model says the majority of soils in this region will fall within this bar. We do the same thing for our soil health management outputs and our baseline system outputs, and we get sort of a set of three bars, which hopefully you are start drawing links with the first slide I had on benchmarking, where we're comparing, aggregate stability for one specific soil texture under three different managements. And then we convert that into a graphic that shows the farmer's measured values relative to the model outputs. So this is an example from a farmer report. The red line here represents that farmer's measured aggregate stability, in this case 0.63, relative to what that aggregate stability would be for soils within that region with the same texture. So now we're making an apples to apples comparison for the ranges of potential aggregate stability given the constraints of the inherent property of texture on that soil. So in this case, this farmer's line intersects the upper end of that blue bar, which represents, their aggregate stability is on the higher end of what we would expect for a soil health management system. So their aggregate stability is looking relatively good compared to what we would expect to see in a baseline system where no soil health principles are adopted. Similarly, right, there's a bit of a gap between that measured value and what we would expect for a perennial reference site in that graph. So there is room where that aggregate stability could increase if they moved into maybe a more perennially focused system or a system that's maximizing soil health. Doesn't mean it's ever going to get all the way to intersecting that green bar, but it shows that there is some potential for that soil to increase its soil health via aggregate stability. So given all that, we can take those modeling outputs and compile them, and we do this in a couple ways. This is an example of the farmer reports we delivered in 2024, showing, for an individual soil sample, you'll notice that each of those essential soil health indicators has a graphic, and that's the same thing we showed on the last slide, where we're comparing the benchmarking modeling outputs to a measured value from that farm, and this grower can then make inferences based on that. So looking at this farm, we see that the red lines representing that grower's measurements all tend to intersect with the green bars of our reference systems, with the exception of aggregate stability. So if I were to say, put on my practitioner hat and say, oh, what is one thing you might want to focus on this farm is thinking about how aggregate stability could be improved. You're doing great. Everything's really actually quite healthy. It's looking like we would expect for a perennial system, but the aggregate stability level is the lowest here. You might want to consider practices that could promote aggregate stability. In this case, I think this is a perennial pasture. So, I'd recommend, maybe minimizing some of the traffic. If you're doing a lot of cutting of alfalfa, that could be, affecting the aggregate stability, or maybe they're applying excess manure that, is causing the something with the salts in the manure to disperse those aggregates. So there's potential reasons here, but in general, you could say this grower is doing really well based on this one soil sample from that field. We also bundled this information in we call regional reports. So we have a handful of these reports out currently, with more on the way. But these describe sort of, for a big geography, right, an entire USDA major land resource area, what the sort of state of soil health is based on the data we collected. So we talk about, what types of management systems were sampled, how many samples were collected in each one, how many fields are represented. We talk about how that changes, how soil health adoption has changed the soils in that region. But I think one of the most useful outputs of that is we include a table in each of these regional reports that describes our essential soil health indicators for, representative soils for that region. So in this example of the Texas Blackland prairies, the soils are dominated by clay loams and clays. And so we look at our benchmarking results and we can say, okay, for a typical baseline soil, organic carbon concentration of 1.1 is a typical value. But if you're in a soil health system, you're looking closer to 1.2. So a grower can take this table, and if they have a clay loam soil, they can understand their soil health tests a little better and get a sense for how they compare to the same soil under different management regimes. And that gives you an indicator of that, relative health of that soil. So we've done this benchmarking work on just over 19 million acres so far. I think this might be a little out of date. I think we have more regions added for this year that are currently being sampled, but that includes, just over 10 million acres in the Cotton Belt, just about 6 in corn, soybeans, and Iowa. So we're really looking to expand benchmarking access to the Midwest. So hoping to develop new projects there to deliver this data. But we also have some data from, things like small grains in Ontario or dairy systems in the Midwest and the Northeast. So one of the cool things about, having these big, large multi-region studies is it lets us understand where our soil health indicators are changing in response to management. So what you're looking at here are our three measured essential soil health indicators in each of the columns. So you've got organic carbon concentration, aggregate stability, and carbon mineralization potential. And then in each of the rows, represents one of the cotton growing regions that we've already completed benchmarking in. Um, and any place you see a check mark shows us where we found a statistically significant difference in our indicator values between baseline systems, so systems without soil health management, and those soil health management systems. Important to note, that the expression of that soil health management systems often changes region to region. Some regions have a lot of cover crops, some regions are largely strip till or no till. And so, when we talk about the differences between baselines in soil health systems, there is a regional effect there. But this is a good illustration of how our indicators or the sensitivity of our indicators to management might be affected by region. So in each of those places where you see a check mark, we saw a statistically significant difference in that indicator between baseline and soil health systems. So in some regions, all three indicators were changing when folks adopted soil health management systems. In some places, only one indicator changed. So example, the Gulf Coast Plain in Texas and the Southern High Plains in Texas, really only had one indicator change. And this highlights why, when we're talking about soil health, it's important to get a full picture and measure multiple things because there's a multifaceted approach here to thinking about soil health and understanding why all of those things could change really is effective in identifying or highlighting changes that might happen. So some places we're seeing bigger changes than in others, and that's why it's really important to measure multiple things when we're trying to answer that question, how healthy is a soil? So with the last couple bit time we have, I don't want to cut too far into Q&A, but I just want to highlight some big picture lessons or takeaways. The first is, soil health is contextual. Just like human health, we need to account for the underlying inherent properties in order to properly assess soil health. So we use the medical, human health analogy with the body mass index. Soil health benchmarking does the same thing, but with soil health indicators and inherent soil properties. So it allows us to account for the natural variation in soil health due to things like climate and texture and drainage class when we're making inferences about soil health related to various management systems. So understanding those inherent properties is essential for making good inferences about soil health because we need to understand the context and the limitations that those inherent properties place on soil health potential. And then, you know, we talked briefly on this, but soil health is multifaceted. We need to measure multiple things to understand or get a full picture of soil health. This applies at various scales, both at the regional scale and at the sort of farmer or individual soil sample scale. So when we look at our regional reports, we need to measure all of those soil health indicators in order to get a complete picture of what's changing in response to management in those sites. And similarly, for an individual grower, it's really important to track how multiple indicators are changing, indicating differences in health in that system. So just a reminder that we need to think about multiple things or multiple measurements when we understand soil health. It's not a single value. We need to measure multiple things in the appropriate context. So with that, I think that kind of wraps up. We have about 15 minutes for questions. Shannon, are you here?
I am. Yep. Jason, thank you very much for your webinar. So we'll start with questions that came in the chat during your presentation. And I'm just going to go in the order that I've got them on my document here.
Great.
Were there any surprises in what factors you selected as key indicators of soil health? And then the second part of that, which I can repeat if you need to later, Did you end up omitting a factor that you thought would be in the final running?
Yeah, so I'm going to assume this question is referring to the NAPE experiment
The
when we were
measurement.
selecting
Yep.
essential soil health indicators. Um, I was not that surprised at what we found based on, my understanding of the literature. Um, what I would say is maybe what surprised me most from that work was that sort of last filter when we were talking about indicators that are usable at scale. And I think this is something that constantly surprises me, first coming from maybe a more academic background and now in a more practical space. The things that work in university research labs aren't always the kinds of things you can get to work in a commercial soil testing lab. So that last filter of understanding and selecting indicators that could be run at commercial labs at scale was I think eye-opening more than anything about, so using aggregate stability as a quick example, there are really good measurements of aggregate stability that we ended up filtering out just because they're too labor intensive for most labs to do at a price point that makes sense for soil health testing, which is why we ended up with using our recommended method of image analysis for slaking as the good indicator of aggregate stability because it's scalable. And so I think for me, the questions that were interesting were those ones related to the practical side of getting soil health testing in the hands of lots and lots of growers means selecting things that are commercially viable and easily done at scale. So that was maybe more surprising than anything. I don't know if that answers the specific question, but I think that was a change for me when it comes to understanding, when we talk about soil health, the practicality of it is really important. If it's not usable, it won't get used.
And speaking of slakes, the next question is related. Are all aggregate stability measures subject to the same texture impact, or is this unique to slakes?
They all have some effect on texture. So aggregate stability, regardless of how you're measuring it, is going to have some impact on texture, which is why it's really important to understand texture when we're making comparisons around soil health. Again, it's all relative, and we want to control for that effect. I will say, once you get down to, soils that are less than 10% clay, most aggregate stability measurements struggle. And most human beings struggle to find aggregates in soils that are 90% greater sand content because the aggregates are so weak and so small. So yeah, they're all affected to some degree by texture, which is why accounting for texture in outputs is really important. A lot of the foundational work and understanding aggregate stability and how it changes with management is all done on replicated trials like the ones we explored with Nation. So these are places where it's the same soil managed under multiple, different management practices, but for all intents and purposes, you're comparing apples to apples because you're always measuring the same soil. When we move to trying to make inferences, across big geographies like major land resource areas, we can't do that same level of experimental replication and control. So we have to find a statistical way to account for that. And this is where I think we often see, it's important to understand when we're comparing literature that's measured on replicated trials to literature where we're doing work on, large spatial scales that we're not always able to make the same types of comparisons or strict experimental controls. Um, so we have to account for texture in other ways.
And then this next one, is there a higher resolution map of the soil health sampling groups available?
Yes, we have one. So if you're interested in it, I can certainly share some of it with you. We're trying to figure out the right way to host it. All of the details and how we do that work. There are a couple of YouTube videos on our website that talk about soil health sampling groups. We've also submitted those methods to the Soil Science Society of America journal. It's in review right now. So once that paper is live, we're going to push to get the data either hosted through the societies or through the data commons. So it will be available shortly, but if you need it today, let me know and we can get you the right kinds of data.
And then this next one, I know my soil type. NRCS made an aggregate stability score, and I have a Haney test. Can I figure out your benchmark?
That's a great question. I would it first would depend on if you're in one of the geographies where we've completed benchmarking. Um, So that's one of the limitations. And things like USDA efforts now to understand soil health also require a lot of data on the back end to get the regional context. So if you are in one of the regions where we've already done benchmarking, um, if you go to our website and you search benchmarking and you filter by documents, you'll find those regional reports and you can compare your outputs to that. I will say the other thing we are working on right now is a web portal interface where folks can log in and submit a sample through a partnering laboratory and get benchmarking results delivered in the same format to them. That system is not live yet, but hopefully in the next few months we'll be piloting that. But in the interim, I would first direct anyone to places where we've already published those regional reports as a starting point, um, and then think about joining in with the SHI portal once that's live.
I think you may have answered the next question, but I'm going to ask it anyway. Who is the person we need to contact to help us find labs to do these soil health assessments?
Yeah, I will have Shannon, when we send out a follow-up email, we will send everyone Liz Reeke's email address and they can email our lab partnership lead, Liz Reeke, who can direct them to the right laboratories.
Yeah. You said you're looking to expand in the Midwest. Is Northern Missouri an area of focus?
Yeah, we would love to partner. So the way, you know, one of the things we're constantly trying to do is find the right regional and local partners who want to collaborate on benchmarking work. So if you are in, that part of Missouri, you have access to farmers and you want to work with the Soil Health Institute on developing a benchmarking project, feel free to reach out to us and we could talk through sort of what it would look like for us to do that. I mean, my goal would be to have benchmarking available everywhere we have row crop agriculture in the US. It just takes time and resources to get those data. And that's, we're exploring, multiple opportunities to do that. So yeah, feel like if you are really interested in gung ho on putting together a benchmarking project, we could talk. So feel free to email me and we can work through, what that might look like.
And then, what was the minimum number of years a field needed to be under cover cropping to be selected for the study, given that parameters such as soil organic carbon and aggregate stability require longer periods to show measurable changes?
Wow, that's a great question and something I just now I'm realizing I didn't highlight. So I appreciate the clarity. So, for everyone on the call, the context here is really important. When you adopt a soil health practice, your soil doesn't change overnight, right? Just because I planted cover crops doesn't mean now I have 2% more carbon in my soil. It takes time for that carbon or that aggregate stability to change. We typically try to look for in our reference sites, places that are under that reference condition for 10 years. And in the soil health management systems, we try to shoot for places that are under a minimum of 5 years under soil health management in order to be classified in those systems. When we actually go out and sample with a grower, we conduct a sort of interview where we collect management history for their previous 5 years. And that helps us understand which category they fall into, whether that's a baseline or soil health management system. But there's also a regional effect here. In some geographies, the prevalence of things like cover crops just aren't that high. And so we might have growers who are considered part of the soil health management group who've only done 1 or 2 years of cover crops, but have done 5 years of strip or no-till. So it depends on the region as well. So it's a little fuzzy, but generally we would love to have every grower who we've sampled in that soil health management category as having 5 years of practice adoption. But that's a great question. An important piece to understand here is that timescales are critical.
We do need to wrap up this webinar, and we'll try to answer anything else in a follow-up email. But Jason, there's one more that I think will be quick to answer. Why are references not established in soils under native vegetation?
That's a great question. Largely, we have a difficult time finding enough native systems in a lot of our row crop areas. So you think about, Iowa is a great example of this. Iowa was all, especially eastern Iowa was all tall grass prairie, and there's like less than a fraction of a percent of the original tall grass prairie left. And those sites tend to be not randomly selected. So if there's a remnant prairie left, it was left aside probably for some other reason, likely that it was unsuitable for agriculture and other reasons. So one, we have a difficulty finding intact native systems. So often those perennial systems are easier for us to find access to. And then lastly, it's difficult to get away from the endogeneity of whether something is still under native systems versus whether it was suitable for row crop systems in the first place. So those two factors really are driving that. But that's a great question. I would love there to be enough tall grass prairie left in the Midwest for us to sample references on tall grass prairies, but just not there.
Great. Thank you so much, everyone, for tuning in today. And this has been a great discussion. We wish we had more time, but like Shannon said, we'll try to respond to the remaining questions on our follow-up email. And thank you, Jason, for taking the time to join us today for this, and to join us today and last week for the two-part webinar series. A short Zoom survey will pop up once this webinar concludes, and we would appreciate if you can provide us feedback. And for those wanting CEU credits, now is a good time to open the CCA app on your phone and be ready to scan the code by selecting Scan Course Code. And I see the code is already up there, so thank you for that. So take your time. We have a couple minutes for you to do that. And please keep a lookout for our follow-up email. Like I said, we're gonna send the webinar recording and other resources. And if you have any questions, feel free to reach out to us and our contact information. Well, you can email Shannon too. Our contact information was there before, but if not, Shannon will communicate with all of us. And be sure to reach out or to check out, sorry, the Farmers for Soil Health website, and you can learn more at farmersforsoilhealth.com.